How do you go about your daily business when you don't know how the kidneys work? So first and foremost, the kidneys are composed of millions of these tiny structures called nephrons. And today I'm going to be going through the structure of a nephron and the different processes that happen in a nephron. So first and foremost, blood is moving through the renal artery, okay? It goes to something called the glomerulus. The glomerulus is a densely packed ball of capillaries that's enveloped by something called the Bowman's capsule, okay? The blood is coming into the glomerulus at a high pressure because the arteriole that brings blood to the glomerulus has a wider diameter than the arteriole that moves blood away from the glomerulus. Okay, the afferent arteriole brings it in, the efferent arteriole brings it away. So because it's coming at such a high pressure, this forces glucose, urea, amino acids and other small molecules out of the gaps in the capillary endothelium through something called the basement membrane and into the Bowman's capsule, forming the glomerular filtrate. Okay, and then this glomerular filtrate moves along the nephron and useful substances are reabsorbed along the way, and the non-useful substances are excreted through your urine. So essentially there's two parts of the nephron. So you've got the cortex, which is where this Bowman's capsule is located, and you've got the medulla. Right, now what happens is that we encounter the proximal convoluted tubule. This is in the cortex. Right, so this is where selective reabsorption occurs, and it's fundamental that you say selective reabsorption because absorption already occurred in the digestive system, in the small intestine and all that stuff. So you have to say reabsorption. Okay, so in the epithelial cells of the proximal convoluted tubule, glucose is actively transported in. Okay, that's why they have loads of mitochondria for ATP synthesis. They've got microvilli, which increases the surface area, so increased rate of diffusion. They've got lots of carrier proteins, channel proteins. Okay, what else? There's sodium potassium pump, which actively transports 2 potassium ions into the epithelial cell and 3 sodium ions out of the epithelial cell. This creates a steep concentration gradient of sodium ions, which enables obviously co-transport to occur. Water is reabsorbed via osmosis because the water potential in the blood is lower than the water potential in the glomerular filtrate, so it moves into the blood. Right, now we move into something called the medulla, and this is called the loop of Henle. The loop of Henle is responsible for maintaining a sodium ion gradient and a water potential gradient. Essentially what happens is that there's two distinct limbs: the ascending limb, which is impermeable to water, and the descending limb, which is permeable to water. So sodium ions are actively transported out of the ascending limb into the medulla. This creates a high concentration of sodium ions in the medulla. Okay, so the water potential of the medulla is reduced. Water then moves via osmosis from the descending limb into the medulla via osmosis. So osmosis enables this to occur, and essentially what happens is that then this enables water to be reabsorbed into the blood. Okay, the longer the water potential gradient is maintained for, the more water is reabsorbed via osmosis. Sodium ions also diffuse into the descending limb, and this whole process of water reabsorption. This causes water to leave the distal convoluted tubule and the collecting duct via osmosis. Okay, and the distal convoluted tubule is located in the cortex, and the collecting duct is located in both the cortex and the medulla. And this is why desert animals have a longer loop of Henle, so that their water potential gradient can be maintained for longer, and they can reabsorb more water via osmosis. And that, ladies and gentlemen, is all you need to know about the kidneys. Thank you for watching.